Hello, I'm Mehmet Hakan Kocoglu. I'm a faculty member at the University of Maryland, Baltimore, and I'm the clinical director for the CAR T-cell treatments for multiple myeloma. Today, I'd like to discuss with you a clinical trial that we've been conducting for an allogeneic CAR T-cell product, P-BCMA-ALLO1. Everything that we're going to be discussing today is publicly available and has been recently presented in attended meetings. As we all know, in the field of myeloma and also lymphomas, CAR T-cell treatments have led to breakthroughs and have been instrumental in the treatment of heavily exposed patients. At the forefront of these approaches are the autologous CAR T-cells, which require collection of the molecular cells from the patients themselves and require a time period that these cells are manufactured through genetic modifications and cell cultures, cytokine exposure, so on and so forth. During this time, there is a great deal of patient attrition because of these are heavily treated patients, and then they do not have a lot of treatment options, unfortunately. And this requires and begs for um, uh, effective bridging therapies and strategies. So that's one of the Achilles heel of the autologous CAR T cells. Um, Now, alongside of the CAR T cells, the chimeric antigen receptor T cells, there's also the bispecific T cell engagers that are off the shelf that they do not require any manufacturing from these patients. However, they also come with some limitations, meaning that they require long-term treatment of these patients, and they significantly increase the risk of infections, especially the ones that are BCMA, B-cell maturation engine targeting treatments. An alternative approach that presents with one and done approach of the CAR T cells, and that is available off the shelf, would have been the allogeneic CAR T cells, and there are several CAR T cells that are being investigated within the clinical trials, both for myeloma and lymphomas. So one such investigational product that we're going to be discussing today is P-BCMA-ALLO1. Um, it's a product that has recently received FDA's Regenerative Medicine and Advanced Therapeutics RMAT designation. The manufacturing of this allogeneic CAR T cell is unique in several different respects, and it uses a proprietary technology. It is a non-viral transposon system, unlike the currently FDA-approved products, which uses lentiviral transduction. And this approach, this non-viral approach, leads to the enrichment of the end product in earlier T cell phenotypes, such as TSCMs, stem cell memory cells, which with the hypothesis that it's going to translate into better functionality, longer persistence, and better expansion of these cells. And there's also a safety switch for these cells, meaning that if there's a toxicity, unexpected toxicity that's been observed, these cells can be decommissioned quite easily. One Achilles heel of the allogeneic products is the tissue incompatibility, right? And there is a risk of graft versus host disease, meaning that once the cells are in the patient, they can recognize healthy tissues as abnormal and attack these cells, which is graft versus host disease, or the other way around. The host's immune system recognizes these cells and tries to take them down, which is a rejection. So to prevent this happening, there are several genetic modifications which are innovative for this product that's been made to these cells. And the final product is enriched through a positive selection. So this study is a phase 1 study. that we are part of, along with more than 10 other US sites. It's an open label study that includes dose escalation of the cells gradually. Um, the patients that are enrolled in this clinical trial are patients who have been treated with more than three lines of prior treatment and have been treated with proteasome inhibitors and immunomodulatory like agents and CD30 targeting therapies. And not just only that, these are the patients that we call triple class exposed and refractory. The study also included the most recent treatments, such as BCMA, B-cell maturation engine targeting treatments, and also GPRC5D, G protein coupled receptor, uh, 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 class 5D treated patients is were allowed in the study. The primary objective of the study is safety. And we are monitoring closely these patients for adverse effects and trying to determine an NTD. And the secondary objective is the efficacy. The this is done through looking at the depth of response, looking at the overall response rate. The study is innovative in several different respects. As I said, it's one of the main correlatives is about having a better expansion, more functional cells, better persistence, not just through the dose escalation, but also looking at different models of lymphodepletion. In the latest arm of one of the latest arms of the study, the median time to the drug was just one single day, which means that these patients did not require any bridging therapy, which is really has a big impact on the patient attrition. So no patient received an bridging therapy. And again, there was no need for tocilizumab prophylaxis, which some of the investigational trials do offer. And outpatient dosing was allowed through the study. As for the demographics of the patient that have been enrolled, more than 50% of these patients have high cytogenetic risk, and 40% of these patients had extramedullary disease. So these are really high risk patients. And about 50% of these patients had been treated with prior BCMA targeting treatments, either another type of CAR T before or a bispecific treatment. And about 30% of these patients have been treated with both BCMA and GPRC5 targeting treatments. As for safety, the primary endpoint, the cytokine release syndrome was seen in 40%, and there were no grade 3 events. Now, in comparison to 70%, 80% of CRS rates that we are currently seeing in FDA-approved products, this is a significant reduction in a good safety signal. In ICANS was seen in 14% of these patients, and again, there were no grade 3 events. In particular, there were no events for Parkinsonism or cranial nerve palsies. The infections were seen in about 50% of the patients, and 14% of them were grade 3. The hematological adverse effects were similar to what had been seen with other products, such as neutropenia, anemia, thrombocytopenia, and lymphopenia. The overall response rate was 88% within the global group and 100% in the BCMA naive population. In patients who had been refractory to both BCMA and GPRC5D exposure, the response rate were close to 80%, was close to 80%. The product showed good in vivo expansion and persistence and good duration of response in earlier arms. And again, with the escalation of the dose, this is likely to be improved further. More importantly, we and others have seen activity against the high cytogenic risk patients, patients with extramedullary disease, and patients who have been previously treated with BCMA targeting agents and have become refractory. And this is being shown through some correlative studies and gene sequencing. The product is currently being explored via phase 1b study at looking at different cell doses, and there's also a potential for redosing, and long-term follow-up results are being awaited. Thank you.